I will start in just a couple of minutes. Okay, I'll now share my slides. So this is the third session of this course, NMR spectroscopy for structural biology. In the last two classes, we have covered basic principles of NMR, 1D NMR, 2D NMR, the different experiments. From this class onwards, we will be going into the biology. Before that, we'll cover, just to revise the concepts we learned in the previous class, here are some sample questions. So the first question is asking that in a nosy experiment, cross peaks are seen between protons, which are, I'll just wait for a couple of seconds and then I'll discuss the answer. So here the answer is less than 5 angstrom apart. If you remember, nosy is the correlation between protons which are not connected by bonds. It happens through dipolar coupling. So protons that are close to each other in space will show nosy correlations. And the intensity of nosy is directly proportional to 1 by r to the power 6, where r is the distance between the protons. Up to less than 5 angstrom, it is visible. More than that, the intensity becomes so less that it is not detectable. So here an example is shown of a peptide. Two amino acid residues are shown. This is residue I plus 1 and this is residue I. So as you can see, the NH of I plus 1 is showing nosy with H alpha of I, H beta of I, and the two H gammas of I. So in the spectra, how this will appear? So as you remember, the diagonal peaks are representative of the one-dimensional spectra. So if you draw perpendiculars from the diagonal peaks, you can see the NH of I plus 1 and the H alpha of I are showing a correlation. Then NHI plus 1, H beta I is showing another correlation. And again with the NH with the two H gammas. So this is how the nosy correlations will appear for protons that are close to each other in space, and the distance has to be less than 5 angstrom. So the correct answer is option A. The next question is asking that in 1H13C experiment, which we saw in the previous class is called HSQC, which nuclei appears along which frequency axis. So here the correct answer is option A. So if you remembered HSQC, it shows correlations between carbon and hydrogen, and the 13C is generally along the F1 axis and the 1H is along the F2 axis. So that is your answer A, that 1H appears along the F2 axis and 13C along F1. Other heteronuclear experiments can be between 1H and 15N as well. That is the more commonly used experiment. And there again, the 1H is always along F2. The other nuclei, be it 13C or 15N, will be along F1. So the correct answer is option A. The next question is asking, in 3D experiment HNCA on a polypeptide chain, correlations are seen between the following atoms of consecutive residues. So the residues are I minus 1, I and I plus 1. And the question is asking that in an HNCA experiment, correlations are seen between which atoms of these residues. So let us look at the HNCA experiment. Here, again, a polypeptide chain is shown. From one H to the next CO is the residue I, and from the previous NH to the CO is the residue I minus 1. In the HNCA experiment, the magnetization starts with the amide hydrogen of the I residue. It is then transferred to the amide nitrogen, and then it goes to the C alpha of the same residue as well as the previous residue. That is why it's the HNCA. H for this hydrogen, N for this nitrogen, and Ca for the two C alphas. So the answer here is H, the amide hydrogen of the I residue, this one, the C alpha, I and I minus 1, these two, and the Ni. So the correct answer is option A. In the next experiment, the same thing is being asked for the HNCO experiment. So in the 3D experiment HNCO on a polypeptide chain, following correlations are seen between atoms of the three consecutive residues I minus 1, I and I plus 1. So here again, similar to the previous case, it starts from the amide hydrogen of the I residue. Magnetization is transferred to the amide nitrogen, and then it goes to the carbonyl carbon of the previous residue. So H for this hydrogen, N for this nitrogen, and CO for the carbonyl carbon. So among these options, option B is showing, so the amide hydrogen is represented by the N superscript. So this is the amide hydrogen of the I residue, this one, nitrogen of I, this one, and carbon with an oxygen superscript refers to the carbonyl carbon of the I minus one, that is this one. So the correct answer is option B. The next question is asking, similarly for the 3D HNN experiment, on a polypeptide chain, the correlations are seen between the following atoms of the three consecutive residues, I minus 1, I, and I plus 1. So here, if you remember, the HNN experiment is used when the C alpha chemical shift separation is not very good. So we don't, instead of using carbon as the third channel, we take two channels as nitrogen and one as hydrogen in the 3D experiment. This is especially used in intrinsically disordered proteins. So we start, the first part is similar to that of HNCA. We start with the, let me just write out the residue numbers. This is I. This is I minus 1. And here if we had another NH, that would have been I plus 1. So the initial part is similar to HNCA. From the amide hydrogen of the I residue, it goes to the amide C alpha of I and I minus 1. C alpha I and C alpha I minus 1. So up to here, it is similar to that of HNC. Here is where the difference begins. From C alpha I, it goes to Ni and Ni plus 1. So from C alpha I, it goes back to Ni. This is already there. This is a new pathway that from C alpha I, it goes to N of I plus 1. And from C alpha I minus 1, it goes to Ni. That pathway is already there. And N of I minus 1. So this is again a new pathway. And from the corresponding N's, it goes to the corresponding hydrogen. So N I minus 1 to Hi minus 1. Ni to Hi is already there, and Ni plus 1 to Hi plus 1. So ultimately, if you look at the options, all the options B, C, and D have some carbon. But in HNN, no carbon is being detected. It's only the hydrogen and the nitrogens. So if you look at option A, the amide hydrogen of the ith residue, that is what we start with, and the nitrogens of I minus 1 and I plus 1, that is shown here. So the correct answer is option A. The next question is also about the HNN experiment. It is asking that the peaks in the plane at F1 have the following signs in a general case where there is no glycine or proline, because if you remember, proline, it is a tertiary amide, and so there is no NH, and glycine shows some anomalies. So if we consider three residues in the sequence I minus 1, I, and I plus 1, and none of the three residues are glycine or proline, so what will be the signs in the plane at F1 of the HNN experiment? So again, when we look at this experiment, so there are three frequency axes. One is hydrogen and two are nitrogens. If we take the plane at a particular value of F1, that is this plane, where F1 at a particular value is taken, the vertical plane, and the change in F2 and F3 is observed. In this plane, what will be the signs? So you can see the three cross peaks in this plane are between the amide hydrogen with the of the I with the that amide nitrogen itself, that is this one, amide hydrogen of I and amide nitrogen of I, then amide hydrogen of I minus 1, amide nitrogen of I minus 1, and similarly for the I plus 1. So these are the three cross peaks that are seen over here. And the signs of these, if you remember the self peak, that is the I peak, will always have positive sign, and the cross peaks, that is with the I minus 1 and I plus 1 residue, will always have negative sign. Here the blue color is shown for the positive and red for the negative signs. So if there is no glycine where the sign pattern can change, this will be the sign pattern. And so here, if you see, the first peak is the self peak. So that will be positive, that is option C. And the next two peaks are the cross peaks, those will be negative. So option C is the correct answer. So now we have covered some revision questions on the topics discussed in the previous class. So now we'll enter the topics of this class. So far we have discussed about NMR. Now we'll slowly go into the biology portion. So the central dogma in molecular biology basically states that from DNA we can get RNA, and from RNA we can get the proteins which are responsible for all biological functions. So DNA is the hereditary material which is passed on through generations, and proteins are the biological machines which are responsible for all the functions. DNA is basically a polymer consisting of 10 to the power 9 monomers, and these monomers are called the nucleotides, which we will look into in the coming slides. And the total length of DNA can be up to 10 square centimeter, but it fits into a cell which is of a much smaller dimension. So the DNA greatly folds into itself. And this is the central dogma of molecular biology, that from DNA through a process called transcription, and RNA is generated, which is called mRNA or messenger RNA. And from this RNA through a process called translation, the proteins are formed. So how the basic concept of translation is that, so RNA, you can imagine it as a single stranded DNA, and instead of thymine, again, we look into the bases in the coming slides, one of the bases is changed to, like instead of thymine, you have uracil. We look into this more eventually. But the main point to remember is three base pairs correspond to one amino acid. So like this, consecutive triplets of base pairs correspond to different amino acids, and like this, the entire protein is generated, and protein is basically a chain of these amino acids. So this is the central dogma in molecular biology. If we look at this in another way, you have the genome, that is the set of genes that an organism has, and that there can be more than 30,000 genes. From there, transcription leads to the transcriptome, that is each gene is forming one mRNA. So there are more than 30,000 mRNA as well. From transcription, from these mRNA through translation, you have the proteins. And now the number greatly increases. There can be more than 100,000 proteins because there are different post-translational modifications. And from each protein, they will carry out its biological function, and that can generate more than 40,000 endogenous and exogenous metabolites. So there are a lot of molecules involved in this entire process. And so now we look at the basic unit of DNA, or the monomeric unit, which is called the nucleotide. It consists of a 5-membered or pentose sugar, a phosphate group, and a nitrogenous base. We will look into each of these components separately. And here, if you look, one particular nucleotide is shown, and nucleotide has a certain direction. So from top to bottom, this is called the 5' to 3' direction. And why it has been named this way is that the top oxygen is called 5' and the bottom oxygen is called 3'. So nucleotides have a certain chain direction, uh, which is important to remember. So here you have the 5-membered or pentose sugar. And so the atoms have been labeled in this way: C1', 2', 3', 4', and this is O4'. To C3', there is a OH attached which connects to a phosphate, and to C5', there is another OH. To C4', there is a C5' attached, to which there is another OH which connects to another phosphate. And this is what, so from this 5' to 3' is the direction of the chain. You can see in the previous slide, 5' to 3'. So, and you can see each phosphate, they connect to the next. So phosphate, through one oxygen, it is connecting to this 5' CH2, and through the other oxygen, it is connecting through, connecting to the 3' end of the next pentose sugar. This bond is called a 3' to 5' phosphodiester linkage, which basically connects each nucleotide unit. In RNA, the difference is, so in DNA, 2' has 2 hydrogen atoms. In RNA, the difference is that this 2', there is one H and one OH. Now for the nitrogenous bases. So there are 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. In RNA, instead of thymine, you have uracil. So instead of T, you have U. So if you look at this, this is RNA. So instead of T, you have U. If you look at the structures of the nitrogenous bases, there are 2 main structures. One is a fused 5-membered and 6-membered ring, which is called a purine, and adenine and guanine fall within this class of nitrogenous base. And another is a 6-membered ring, and within this class, you have cytosine and thymine, or uracil if it is RNA. And the difference between uracil and thymine is that thymine has an additional CH3 group at the carbon labeled 5. So you can remember the numbering of the nitrogenous bases, so that will help in when we are looking at the NMRs later. Now comes the concept of base pairing. So base pairing is very specific, and adenine forms 2 hydrogen bonds with thymine, and guanine forms 3 hydrogen bonds with cytosine. So A can only pair with T, and G can only pair with C. So only certain bases can form base pairs, and 2 strands come together to form a double helical structure, which is the DNA structure. You can see A and T have 2 hydrogen bonds, and G and C have 3 hydrogen bonds. Now we will look more into the sugar ring. So this is the pentose sugar ring, and there are 5 bonds. So the rotation along each bond can be depicted by a torsional angle. Here they have been depicted as nu0, nu1, nu2, nu3, nu4. If you remember, torsional angle basically is the angle between 4 atoms, or if you consider 2 planes like this, it is the angle between these 2 planes. That is called a dihedral angle or a torsional angle. And these 5 torsional angles of the pentose sugar are dependent on each other, since the ring is a closed structure. So if you define one angle, that will automatically fix the other 4 angles. Now this sugar ring can have different geometries. The most common geometry is where 3 atoms are in a plane, and one is above the plane and one is below the plane. So just one second. So if you see here, the 1', 4', and let me just write this. So this is 1 prime, 1 prime, 4 prime, and 5 prime. Sorry, the 5 atoms in the ring. So 1 prime, 4 prime, and this is C4 prime. They are all in the, let me write this also. This is 4 prime, C4 prime. So C1 prime, O4 prime, and C4 prime are in the plane. 2 is above the plane, 3 is below the plane. So this entire column of compounds follow the same basic principle that 3 atoms, 1 prime, O4 prime, and C4 prime are in the plane. 2 is above the plane, which is called endo, and 3 is below the plane, which is called exo. This group of conformations have been termed as south or S conformations. The other kind is the north or N conformations, where again the 1 prime, O4 prime, and C4 prime are in the plane, but here it is opposite. 2 prime is below and 3 prime is above. In south, 2 prime was above, 3 prime was below, and in the north conformations, it is the opposite. 2 prime is below and 3 prime is above the plane. Now there is a certain formula which can define each nu. So nu I, where I can vary from 0 to 4, is tau m, where tau m is the maximum distortion of a particular atom from the plane of the ring. So if this is the plane of the ring where the 3 atoms are there, how much a particular atom can be distorted from this plane is the tau m, the maximum value. So it is typically around 38 degrees. Then there is a cos in the bracket P. P is defined as the pseudo rotation angle from 0 to 360 degree, plus I minus 2, I is this I, whether it's 0 to 4, into delta. Delta is a constant, that is 144. So using this formula, the different torsional angles can be predicted. Now DNA can be of 3 major conformations. The most common form of DNA that we normally, that occurs in most cases is the bDNA. It is the most elongated DNA. There are around per base pair, there is around a rise of 3.4 angstrom, and in each turn of the helix there are 10 base pairs. In this structure you can see there are 2 kinds of grooves forming. One is a major groove, which is a larger groove, and one is a minor groove, which is a smaller groove. And most protein DNA or metal DNA interactions occur in these grooves. This is a right-handed helical structure. Another such right-handed helical structure is aDNA. You can see clearly aDNA is broader than the bDNA. And also the base pairs are tilted with respect to the axis. So if you consider the axis like this, and here also if you consider a straight line, in bDNA, the base pairs are much more perpendicular to the axis, but in aDNA, they are quite tilted. And mostly RNA occurs in this form. So DNA is very rarely found in this form, but mostly you will find RNA in this conformation. The most rare kind of DNA is called zDNA, and it does not form a proper helical structure. It is kind of a zigzag structure. You can see these two are very clearly helices, but this is not. And this is a left-handed structure. Here, the repeating unit is actually a dinucleotide. It is not a single nucleotide like A and B DNA, and these are rich in CG segments. So zDNA is the most rare kind of DNA. If you look at the different like aDNA, bDNA, and zDNA, they differ a lot in the different torsional angles. So here is the pentose sugar ring. This is the phosphodiester bond, and this is the bond to the nitrogenous base. So different the rotation along different bonds have been termed by these letters. So if you can see, alpha is the rotation between phosphate and O5 prime. Beta is between O5 prime, C5 prime. Then C5 prime to C4 prime is gamma. C4 prime to C3 prime is delta. Delta is also nu 3. Basically, it is within the sugar ring. Then C3 prime to O3 prime is epsilon. O3 prime to the next phosphate is the eta. And chi is basically between C1 prime and the nitrogenous base. So these are all the different torsional angles, and they vary quite a lot between the different DNAs. You don't need to remember the values. You can just remember that the delta angle represents the sugar ring. You can see the delta, it is clearly within the sugar ring. And aDNA has, in aDNA, the delta angle is C3 prime endo, and the bDNA is C2 prime endo. So the delta angle. You can see aDNA and bDNA. So C3 prime endo was the north conformation. So aDNA has the north conformation and bDNA C2 prime endo, so it's the south conformation. Whereas zDNA has alternating values. And the chi angle represents the orientation of the base with respect to the sugar ring. That is this chi torsion. Now the base pairs themselves can be oriented in different ways. We have already seen that in the base pair, one side has the minor or smaller groove, and the other side has a much larger or major groove. The most common kind of base pair orientation is where the base pair is perpendicular to the helical axis. This is the coordinate frame. From this, the base pair, so we are consider the helical axis is Z and the base pair is in the XY plane. Now the base pair can tilt along Y axis. That will be called the tip. If it tilts along the X axis, that will be called inclination. Now the base pairs, instead of being perfectly connected like this, there can be a certain opening between them. Or they can be rotated with respect to each other. That will be called the propeller. Or they can be bent with respect to each other. So that is called the buckle. Now, the third column is the relative orientation of two consecutive base pairs. If they are twisted with respect to, they can be twisted with respect to each other. They can be, so here two base pairs are bent in such a way that they are opening up along this axis. This is the x-axis. That is called roll. Or if they are opening up along this axis, this is the y-axis. That is called tilt. So these are all different relative orientations of the base pairs. Now again, we'll go back to what we saw, the different kinds of DNA. Here we are mainly comparing bDNA and zDNA. In bDNA, the most common kind of DNA, two right-handed helices run in opposite directions. By opposite directions, it means that if, one second, if this strand is 5' to 3', the other strand will be 3' to 5'. And the base pairs are well stacked. We saw in the previous case also that the base pairs are almost perpendicular to the axis of the helix in bDNA. And in zDNA, there is kind of a zigzag conformation. It is not as symmetric as bDNA. And the base pairs are not very well stacked with respect to each other. So you can see stacking of base pairs is not very good. Here the base pairs are pointing outwards. Here they are pointing outwards. So stacking is not very good in zDNA. Another thing is bDNA is a right-handed helix and zDNA is a left-handed helix. Now if we look at the residues per turn, bDNA has 10 residues for one complete turn of the helix, while zDNA has 12 residues. And since the monomeric the repeating unit is a dimer, there are 6 dimers. Diameter of bDNA is around 20 angstrom, zDNA is around 18 angstrom. Rise per residue is slightly higher in zDNA than bDNA. Now we'll define a term called pitch. If the height is the total height of the helix. Pitch is the rise per turn. For a single turn of the helix, how much is the rise, that is the pitch. So now if we look at the pitch, so rise per residue is 3.4 angstrom for bDNA, and there are 10 residues per turn. So the rise for one turn will be 3.4 into 10, that is 34 angstrom. Similarly for zDNA. Now if you look at the rotation per base pair, rotation per base pair for bDNA is 36 degrees, and there are 10 residues per turn. So as expected, rotation per base pair into the number of base pairs per turn will complete an entire 360 degrees. So 36 into 10, 360 degrees. And similarly for zDNA, rotation per base pair is around minus 30 degrees into 12 base pairs per turn. So that is also giving you the complete turn of 360 degrees. So like this, bDNA and zDNA differ in various parameters. We will define the other parameters slowly. So one parameter is the glycosidic torsional angle chi. We have already talked about chi earlier, where chi is the rotation between this carbon of the pentose sugar and the nitrogenous base which is connected to it. So there can be two major conformations of chi. One where the 5-membered ring of the nitrogenous base, this 5-membered ring, is on the same side as the oxygen of the sugar ring. Then it is called the anti-conformation. And if this 5-membered ring is on the opposite side to this oxygen in the sugar ring, then it is called a syn conformation. Here you can see the example shown in 3D. This is the 5-membered ring of the nitrogenous base, and it is towards this oxygen of the sugar ring. This is the anti-conformation. And here the 5-membered ring is facing away from this oxygen. That is the syn conformation. So this angle is important in defining the orientation of the nitrogenous base with respect to the pentose sugar. Now we talked in one of the first slides that the length of DNA is in the order of centimeters. So how is this accommodated in the tiny nucleus of the cell, which has much smaller dimensions? And that lies in the folding of DNA. Now we will look at how the different nitrogenous bases form pairs. So the most common kind of pair that we have seen, which occurs in bDNA, is where A forms with T or U, 2 hydrogen bonds, and G forms with C, 3 hydrogen bonds. This is called Watson-Crick base pairing, and it is what we have seen so far. Now, this is not what happens in 100% of the cases. There are different other base pairs that can form, which I will now discuss. Now, if we consider an axis like this shown by the dotted line, and the U or the thymine residue is rotated around 120 degree around this dotted line. That means this NR, which was pointing down in the Watson-Crick base pairing, is now pointing up. So just imagine it is flipped by 180 degree along this line. So the U is flipped like that. So then it is called reverse Watson-Crick base pairing. Similarly, if the C is flipped by 180 degree along this line, so the NR, which was pointing down, is now pointing up. Then instead of 3 hydrogen bonds, 2 hydrogen bonds are forming between G and C, and both of these are called reverse Watson-Crick. There is another totally different class of base pairs that can form, which is called the Hoogsteen base pairing. Here, one NH of 5-membered ring. So here you can see in the fused 5 and 6-membered ring, only the 6-membered ring was participating in hydrogen bonding in both Watson-Crick and reverse Watson-Crick. In Hoogsteen, one NH from 6-membered and one NH from 5-membered participates in hydrogen bonding. Same pairs form. Here also AU was forming, here also AU is forming. But in Watson-Crick, both the NH was coming from the 6-membered ring, whereas in Hoogsteen, one NH comes from 6-membered ring and one comes from the 5-membered ring. And you can similarly have reverse Hoogsteen. So in reverse Hoogsteen, this U is flipped by 180 degree. So NR, which was pointing down, now it points up. And then it is called the reverse Hooke's-Steen. And there is a third kind of base pairing where the base pair itself is changed. So from Watson-Crick and Hooke's-Steen, A still pairs with U or T and G pairs with C. In wobble base pairing, the partners change. That is, G pairs with U and A pairs with C. So this is the wobble base pair where G is pairing with U. And again, if you flip the U by 180 degree, that will become reverse wobble base pair, and so on. So instead of the expected AT, you will have AC, and instead of GC, you will have GU or GT. So these are the different kinds of purine-pyrimidine base pairs, where purines are the, one second. Yes. So these are still the purine-pyrimidine base pairs, where one is a 6-membered ring and one is a fused 5 and 6-membered. Now you can have heteropurine base pairs as well, or homopurine base pairs. So you can have A pairing with G. So these are very different kind of base pairings that can still happen. So purine base pairs, you can see purine is the fused 6 and 5-membered ring. So let us first look at the homopurine base pairs. So homopurine base pairs is basically adenine pairing with adenine. They are homopurine because they are the same base pair, and adenine is a purine because it has a fused 6 and 5-membered ring. So it is called AA N1 amino because N of one is pairing with the amino of the other. Again, N1 of the other, this is pairing with the amino of this, and it is symmetric. Then you can have again AA N7 amino. So N7 of one adenine is pairing with the amino of the other. N7 of the other adenine is pairing with the amino of the first one. So again, this is symmetric. Then you can again have AA, where this is asymmetric. So N1 of one is pairing with the amino of the other. N7 of the other is pairing with the amino of the first one. So here the pairing is different, and so it is not symmetric. Numbering you can remember shown here. Then you can have GG, which is again homopurine because G is the other purine. And here you can have N1 carbonyl. So N1 to the carbonyl of the other G, and again N1 to the carbonyl of the first G. So it is symmetric. Then N3 amino. So N3 of one G to the amino of the other G, and N3 of the other G to the amino of the first G. This is also symmetric. Then you can have homopurine but non-symmetric. So N7 to N1. So if you look at this, N7 of one G to the N1 of the other G, and carbonyl of one G to the amino of the other G. So clearly this is non-symmetric. Again, you can have N1 of one G to the carbonyl of the other G, N7 of one G to the amino of the other G. So this is also non-symmetric, and these are all homopurine base pairs. Now you can have heteropurine base pairs. So G and A, they are the two purines, but these are different, and so they are heteropurine. Here you can, there is N1 with N1. So N1 of A with N1 of G, sorry, N1 of G with N1 of A, and carbonyl of G with amino of A. Again, AG can form a different kind of base pairs. N3 of A with amino of N3 of G with amino of A, and amino of G with N1 of A. These two are AG. So these were GA, these are AG. And similarly, they have been named according to which are the atoms forming the base pairs. So these are homopyridine and heteropyridine base pairs. You can also have pyrimidine-pyrimidine base pairs, where pyrimidine is the six-membered nitrogenous base. So CC can form a base pair in different ways according to the hydrogen bond that is forming. You can have, so these are, CC is homopyrimidine. UU, this is also UU, this is homopyrimidine. CU or UC, these are heteropyrimidine. So like this, pyrimidine-pyrimidine base pairs can also form, and they are named similar to what we saw in the previous slide. Another is when in CC, one of the C is protonated. So that will become C plus, and normal CC, if you look at these two, they are forming two hydrogen bonds. But when one of the C is protonated, so imagine this nitrogen is protonated, then a third hydrogen bond can form here. So that is where C and C plus form three hydrogen bonds between each other. That is often called the I motif. Now we will look at the different secondary structures that DNA can form. So the most common is the DNA duplex, that is the double-stranded DNA. It can open itself up like this. So you have the double-stranded DNA, and here it opens up into a loop. So in the loop region, it is just a single strand. So this is a single-stranded region called a loop. And you mainly name this as a hairpin loop and this as the hairpin stem. Then you can have what is called a bulge. So the loop is happening at one end. Bulge happens in the middle, and it only involves, it generally involves much fewer base pairs compared to loop. If you have a single base bulge, so only at a single base pair, the pairing is not there and it is single stranded at a single base pair. You can also have internal loops where the sequence is not complementary. So suppose you have A and G. So they are not pairing with each other. They are pointing outwards, and they can instead form pairs with other DNA or some other molecule. This mismatch can happen at a single base pair. They can form at multiple base pairs, in which case this loop will be larger, and they can be symmetric. So three base pairs this side, three this side. That is symmetric. Or it can be asymmetric. Three on one side, two on the other side. So that can also exist. And another more complex structures are where there are three stems. So this is a double-stranded DNA, this is a double-stranded DNA, this is a double-stranded DNA. They are connected by this kind of internal loop structure. So this is called three-stem because there are three regions of double-stranded DNA. Or it can be four-stem. So there are four regions of double-stranded DNA, and they can be connected by this kind of bulge or internal loops. So these are all secondary structures. Now you can have what is called a base triple. So far we were only seeing pairing between two base pairs. But two nitrogenous bases were pairing, so we were calling it base pairs. Now three nitrogenous bases can also pair, and these are generally called base triples. So TAT, CGC, GGC, AAT, these are different ways in which three nitrogenous bases are pairing with each other. And you can see there are arrows shown here. These arrows are basically short distances that are observable in NMR. So nosy, we started this class with a question on nosy, which is when two hydrogens are close to each other, they will show nosy cross peaks. So wherever you can see the arrow, these hydrogens are close to each other and they will show cross peaks in a nosy spectrum. You can also have a tetrad structure where four base pairs come together to form a very stable structure. And one of the well-known tetrads is called the G tetrad, where four guanines come together and to form a very stable symmetric structure. And different G tetrads can form the complexes with each other. So if you imagine each like this square to be 1G tetrad. So there can be 4G tetrad like this, all parallel to each other, or there can be some parallel, some anti-parallel, or there can be loops between them. We'll look more into the details later. So these kind of structures, which are formed from stacking of the G tetrad, are called quadruplexes. So we have looked a lot into the biology. So how does this correlate with the NMR? So NMR of 1H we know ranges from 0 to around 14 ppm, and different protons appear at different ranges over here. So at the most upfield shifted ranges, you have the sugar ring protons. For DNA, they are spread out. So 2, 2 prime occurs most upfield, followed by 4, 5 prime, then 3 prime, then 1 prime. And in RNA, if you remember, the only difference in the pentose sugar is that at 2 prime, where DNA had 2 hydrogens, RNA has 1 hydrogen and 1 OH. Due to this 2 prime proton in RNA, OH is an electron withdrawing group, so it will withdraw electrons, and the 2 prime proton will become more downfield shifted in RNA. That is what is observed. 2 prime in DNA was upfield, and in RNA is quite downfield because of this OH group. Next are protons of the nitrogenous base. So you can see H2, H8, H6, these are all protons of these bases. You can see the numbering here. More downfield shift are where the base pairs and other structures like loops occur. So ATGC for DNA or AUGC for RNA, they occur much more downfield shifted, greater than 12 ppm. Another kind of protons are the imino, that is NH, and the amino, that is NH2 protons. These are exchangeable protons, that is, they can exchange with the solvent. So if your solvent is D2O, the NH2 can become ND2, and D is not NMR active. So then this peak will disappear. So in order to observe these protons, the solvent needs to be H2O. Otherwise, if it is D2O, the protons will exchange with the solvent and we will not be able to observe these peaks. And these iminoprotons are basically where the hydrogen bonding is not there, and they will occur in a lot in loops where you can see it's single strand. Normally, the imino will be involved in hydrogen bonding. But if it is loop where there is single strand, the imino will be free and you will be able to observe it. Here is an example, and thymine has a CH3 group. So this is a very unique group because it does not occur in any of the other nitrogenous bases, and it occurs very much upfield, so less than 2 ppm. So if such a CH3, if there is a peak less than 2 ppm, it is very characteristic of the thymine. Now we will look at some NMRs of DNA structures. So here you have a structure where there is a double-stranded DNA, and at the end there is a loop, and same on the other side. So each loop is formed by 4 cytosines which are not paired, and these cytosines in the loop will not be observable since they are exchanging. We will only be able to observe the bases in the double strand. And the double strand also is symmetric, so we will not see all the bases. So you can see it is symmetric because GGAA, and it is the same on the other. So you can see if you label the base pairs. So we will look at 1 second. Okay, so amino, one thing to remember is that wherever the loop is there, those protons will exchange very rapidly with the solvent, and so we will not be able to see those peaks. In the duplex itself, the amino protons exchange very rapidly with water, and so amino protons may often not be observed. So what we will see are the iminoprotons, that is NH, which occur between 12 to 15 ppm. That is the region we are looking at. Each base pair has a single iminoproton. If you look at this base pair, there is a single NH. Imino is the NH. You can see there are no other NHs. And if you look at the GC base pair, again there is a uh, uh, there is a single NH, because this middle NH is what will be observed. This NH is this is from NH2, this is from NH2. So there is a single NH in GC. So each NH peak is representative of one base pair. So we will see one for we will see G1 and then G13 and G14. So you can see, just one second. So what we can see is that the amino NH belongs to the thymine. Amino NH belongs to the thymine, or the amino NH belongs to the guanine. So we will only be able to observe thymine or guanine, which will be representative of the base pair. So for the guanine, there are three unique guanines, this G1 and at the end G13, G14. These two guanines and 13 and 14 are equivalent, so we will see a single peak for them. And for the thymines also, there are three thymines. This 3 and then 5 and 6. If you look at the other thymines, they will be equivalent. So this thymine labeled 5 and 6 is adjacent to two cytosines, and these two bottom thymines are also adjacent to two cytosines. So 5 and 6 and these two are equivalent, and this thymine 3 is equivalent to this. So the ones which are labeled by numbers, G1, G13, G14, and T3, T5, T6 are the only ones you will see. So that is 6 peaks. So basically you can see in the middle, from bottom to top, it is increasing temperature. You can see G1, G13, G14, three distinct peaks, and T3, T5, T6 are kind of merging, but you can see 3 peaks. So you can see the 6 iminoprotons over here. And as the temperature is increasing, the base pairs will start separating. So they will get more and more exposed to water or the other solvent, and they will disappear till you can see at 303 Kelvin, the base pair peaks have disappeared and there is just a very broad spectra. So with increasing temperature, as the base pairs get more and more exposed to solvent, hydrogen exchange will happen, and so the peaks will disappear. Similarly, let us look at a DNA hairpin. We saw that hairpin is where, just one second. Just one second, okay. Right, so DNA hairpin is where you have a double-stranded DNA, and at one end you have a loop where it is single strand. Now the nucleotides in this loop will not be observed since they are single-stranded and they will exchange with water. The aminoprotons also exchange with the solvent, and so they are very much merged. But what will be clearly seen are the iminoprotons. Now here, In the previous sequence, there was a kind of symmetricity, and so there were only 6 peaks that were seen. But here there is, you will see clearly there is no such symmetricity, and you will see you will clearly see 6 peaks, 1, 2, 3, 4, 5, 6 for each base pair. So you can see for each base pair, one imino peak is seen at 5 degrees Celsius. With increase in temperature, the peaks are kind of merging. They are because they are getting more and more exposed to the solvent, and they are getting broadened. Uh, this is also called melting of the DNA duplex. And here what is happening is that, uh, just one second. So here what is happening, as the temperature is increasing, the first signal to disappear is the CG1, that is this. So the order of disappearance is basically the ones at the end disappear first, because when you are heating it, the first portion of the DNA that will unwind is at the terminal. Then the will be the second, then the third, and so on. So with increase in temperature, you can find out which peak disappears first, which second, which third, and so on. And so you can predict the sequence of the DNA as well by increasing the temperature. So whichever peak disappears first, that is at the terminal, and by the order of disappearing, you can figure out from the terminal to the interior. Now, if the DNA is modified, whether it is stabilizing or destabilizing, that can also be observed. So in the phosphodiester bond, instead of a double bonded O, you can have a double bonded S, which will be called a phosphothioate bond. So there is the unmodified DNA where it is all the phosphodiester bond. There is a DNA where one of, so in each base pair, one oxygen of the phosphodiester has been replaced by sulfur. So you can see at each base pair S is written. So that is the phosphoborothioate modification. Or you can have at each base pair phosphodiester bond, both of these oxygens have been modified to sulfur. That is why 2s is written, because there are 2 sulfur atoms inserted. That is the phosphodithioate modification. Now, if you take either you can plot absorbance with respect to temperature, or you can plot chemical shift with respect to temperature. These are all showing this kind of 2-state transition where the DNA is going from a folded to an unfolded state. And the midpoint of these curves will give you the melting temperature at which the unfolding is starting. So if you look at it, this the melting temperature of the unmodified is around 60. For the modified, where you are having a single sulfur modification, this one, that is around 46, 47, and where you are having 2 sulfur modifications, that is around 20. That is this one. So as you go from 1 to 2 to 3, that is the unmodified DNA, to the single sulfur modification, to the 2 sulfur modification, the stability of the DNA is decreasing because the melting temperature is decreasing. So this modification is destabilizing the DNA. Now we will look at the nosy spectra in DNA. So here, one second. So here a single DNA sequence is shown, and in the nosy spectra, you can see this, whichever peaks appear around this range, they are correlations between this H2, H6, H8 are protons of the nitrogenous base, and this H2, 2' CH3 are protons of the pentose sugar. So this is showing CH3 is for the thymine, that is specific. So this region is showing correlations between the protons on the nitrogenous base and the protons of the sugar ring. This region, and which protons of the sugar ring specifically, 2 prime, 2 double prime, is specifically because it is more upfield. The others are more downfield, so their correlations appear below. So the other protons of the sugar ring, that is 3 prime, 4 prime, 5 prime, so on, they will show correlations with the nitrogenous bases around this region. And this region, which is on the diagonal, is basically for base-base stacking interactions. So if you have like here 2 nucleotides are shown, then one base with the other base, if they are stacking on top of each other, nosy between them will appear in this region. So here 2 nucleotides are shown. We are going from 5 prime to 3 prime direction. So there can be different kind of nosy. One can be, so this is the sugar ring of the I residue. This sugar ring can have nosy with the nitrogenous base of the same residue or with the nitrogenous base of the previous residue. You can see all these distances, 3.5 angstrom, 4.3 angstrom, these are all within the 5 angstrom threshold within which nosy can be observed. So you can have correlations within the same nucleotide or with the next nucleotide. And this intensity will depend upon the distance between these hydrogens. So each base will have, so here again, if we look at the exact nosies that are being observed, this is the H1 prime of the sugar ring. It is showing nosy which with this hydrogen, that is H8 of this ring. So that is around 3.5 angstrom. This is within the same residue. Similarly, H1 prime of the next residue will show with this hydrogen of the nitrogenous base, that is also within the same residue. And between the two residues, that is from the nitrogenous base hydrogen of I+1 to the H1 prime of the sugar of I, around 4.3 angstrom. So this will also be seen. So for this, if we take 2 consecutive residue, 3 nosy cross peaks are expected. So each base will have 2 cross peaks, and if we look within the same base, then there will be, if we look within the same residue, there will be 2 cross peaks. One is with itself, this 3.5 angstrom, one is with the next one, that is 4.3 angstrom. So here it is a DNA sequence. If we start from T8, the self peak is shown in green and the sequential peak is shown in red. So if we start from here, T8 shows self peak, that is this one within itself, and one with the one residue before it. So this is the sequential peak with G7, that is this peak. Similarly, G7 is showing a self peak and a sequential peak with G6. G6 is showing a self peak and a sequential peak with G5. Like this, if you move through the spectra, you will end up in T1. T1 will only have self peak because there is no peak before it. So like this, if you follow the nosy spectra, you will be able to figure out the DNA sequence. Now this is about the sugar puckering, how that can be understood from NMR. So we have seen that the north conformations are where the 3' is endo, that is above the plane, and the south conformation is where the 2' is endo, that is 2' is above the plane. Now, what are the different coupling constants we can have in the sugar ring? So 1' hydrogen can couple with 2' hydrogen. 1', so there are two 2' hydrogen. We will call them 2' and 2 double prime. So 1' with 2', 1' with 2 double prime. That is these two. Then 2' with 3', 3' with 4', sorry, 2' with 3', 2 double prime with 3'. So that is these two. And then 3' with 4'. So these, OH we don't consider because this hydrogen does not exist. It connects to a phosphor. Here it is shown separately, so it's shown OH. But in a DNA, actually, it will be a phosphodiester bond. So this hydrogen will no longer exist. Similarly for this OH. So the coupling constants are only the 3-bond coupling constants that we considered, the 3J. So again, I'll repeat, from 1' to 2', 1' to 2 double prime. 2' to 3', 2 double prime to 3', and 3' to 4'. These J values, they vary with the pseudo rotation angle, which we have described from 0 to 360 degrees. So now we will look at the different structures. For the 3' endo structure, the pseudo rotation angle varies between 0 to 18 degrees. So if we take a vertical, so pseudo rotation angle around 0 to 18 degrees, say we have taken 9 degrees here, we draw the vertical. So wherever this is meeting each of this curve, that is the J value. So each particular J value, so there are 5 J values. How each of these J values change with the pseudo rotation angle P, that has been plotted. For the north structures, 3' endo structures, pseudo rotation angle P varies from 0 to 18 degrees. Let us consider 9 degrees. And we have drawn a vertical from around 9 degrees. So for the north structures, this J value, that is 1', 2 double prime, is 0. You can see so 1', 2 double prime is 0. The other 4, they are around 6 Hz, 6, slightly higher, 7, 8. They are basically greater than 6 Hz. We are just taking them. So again, I'll repeat, at the P values of the 3' endo or north structures, this J value, that is the 1', 2 double prime, is this is 1', 2'. The 1 second. This is sorry, this is the 1', 2'. So again, I'll just repeat it. So at the P values of 3' endo or north, that is 0 to 18 degrees, the 1', 2' J value is around 0 Hz, and all the rest are greater than 6 Hz. And at the C2' endo or south conformation, that is 144 to 162 degrees, we have just considered at 144. Here, this dotted one, that is 2', 3'. No, this dotted, this one is actually 3', 4'. Yeah, this is dashed one. 3', 4' is 0 Hz, and the others have different values, which we have written from this graph. So each of these structures, the 5 J values are different. Now we will look at how the cozy cross peaks will appear for the sugar ring in each of these structures. So here we have plotted 1', 2', 2 double prime, 3', 4'. And similarly on the x-axis, because cozy is hydrogen-hydrogen correlations itself. Now, what are the peaks that will be observed? So 1', 2' coupling is 0. So this will not show any peak. The other 4 will show peaks. So 1' with 2 double prime. 2', 2 double prime appear around the same region. So you will see only one peak here. 1' to 2 double prime is the peak you will see. Since 1', 2' is 0 Hz, that will not be observed. Then you have 1' to, you have 3' to 4', that is this peak, 3' to 4'. Then you have 2' to 3' and 2 double prime to 3', that is these two peaks. So these are the peaks. Out of the 5 possible peaks, you will observe 4. One will be not observed since the coupling is 0. For the south, that is the endo conformations, you have 3 prime 4 prime coupling 0 and 3 prime 2 double prime coupling is very small. It's 1 to 2 hertz. So this is also not observed. For the other three, coupling 6 hertz, 7 to 8 hertz and 10 hertz, these three you will observe. So 1 prime 2 prime and 1 prime 2 double prime. So 1 prime 2 prime and 2 double prime, these two peaks, and 2 prime 3 prime, that is this peak. So these are the peaks that you will observe for the C2 prime endo or south conformations. Now if we look at the fine structure of these peaks, we are looking at the at C2 prime endo. So C2 prime endo, we have to remember that for C2 prime endo is this case. So this case is two of the peaks, their coupling is very close to 0. We are particularly looking at the 1 prime 2 double prime peak. So 1 prime has coupling with 2 prime and 2 double prime. So first 1 prime is being split by 2 double prime, and since this is the active coupling which we are observing, the phases will be different. So it is splitting into plus minus. Then 1 prime will be split by 2 prime. This is passive coupling, so plus will remain as plus and minus will remain as minus. Now we have seen in the previous class that the magnitude of the couplings mattered. The active coupling is around 6 hertz, so this is smaller. Passive coupling is around 10 hertz, so this is larger, so it is crossing each other. So for 1 prime, the signs will be plus minus, that is what is written here. Now 2 prime, it will first be split by 1 prime, that is the active coupling, so the signs are different, it is getting split into plus and minus. Then 2 prime will be split by 2 double prime will be split by 2 single prime. This is passive coupling, so plus remains as plus and minus remains as minus. Now 2 double prime should also be split by 3 primes, but it is very small, so we ignore this. J values are hardly 1 to 2 hertz, so there is no splitting here. Now here again, if we look at the magnitudes, 2 double prime 2 prime is a much higher magnitude, around 14 hertz, because it is geminal coupling, and this one is smaller. So again, there is a crossing that happens. So the sign pattern is plus minus plus minus. So there are 4 components on each side. So we have a total of 16 components with this fine structure. So this is for C2 prime endo, this case. Now if we have C2 prime endo itself, but we were looking at 1 prime 2 double prime. Now if we look at 1 prime 2 single prime. So 1 prime is being split by 2 single prime. This is the active coupling, so the signs are different, plus minus. Then 1 prime is again being split by 2 double prime. This is the passive coupling, so plus remains plus, minus remains minus. Now if we look at the magnitudes, this is greater, around 10 hertz. This is lesser, around 6 hertz. So there will be no crossing. So the sign pattern is plus plus minus minus. That is what it remains for 1 prime. Now for 2 prime, it will be split by 2 double prime. Here it is passive coupling, so both remain plus, because active coupling is 1 prime 2 prime. Next 2 prime will be split by 1 prime. This is the active coupling, so plus goes into plus minus, again plus goes into plus minus. And finally 2 prime will be split by 3 prime, and this is also passive coupling, so plus remains plus, minus remains minus, plus remains plus, and minus remains minus. Now if we look at the magnitudes, 2 prime 2 double prime has the highest magnitude, 14 hertz, because it is a geminal coupling. 2 prime 1 prime has lesser magnitude, 10 hertz, so there is no crossing yet. But now 2 prime 3 prime has smaller magnitude. So here a crossing will occur. Signs of these two extremes, there will be no crossing, but in the middle a crossing will occur, because this is 10 hertz and these are 7 to 8 hertz. So the sign pattern will be plus plus, minus plus, minus plus, minus minus. That is what is written here. So you have 2, 3, 4, 5, 6, 7, 8. 8 components for 2 prime, 4 components for 1 prime. So total 32 components for the 1 prime, 2 prime, peak fine structure in C2 prime endo. Now the problem is that if you have so many components, like 32 components, then many of them cancel out. You can see, you can hardly see 4 components here. You can maybe see 8 here. So a lot of them are cancelling out because there are so many components. So the way out of this is a special technique called COSY 45 or E-COSY, which simplifies the fine structure so that there is not so much cancelling out. So we don't need to understand the mechanism of this. You can just remember this is a technique to simplify the fine structure so that the cancellation is reduced. And if the cancellation is reduced, we can find out more of the coupling constants from the fine structure. So you can see in COSY 45 compared to COSY, clearly many more cross peaks are observed, and so more coupling constants can be calculated. Here an example is shown of COSY 45. So this is the 1 prime 2 double prime peak and 1 prime 2 prime peak. So if we take the difference between cross peaks, we can get different J values. So one J value from here is 5.8 Hertz, one from here is 14 Hertz. We know 14 Hertz comes from the geminal coupling. So that is between 2 prime 2 double prime, and this 5 Hertz is around the what is expected for 1 prime 2 double prime. Similarly, you can get another coupling constant 9.5 Hertz from this 1 prime 2 prime, which appears here for this 1 prime 2 prime cross peak. So for COSY 45 or E-COSY, cross peaks are much more clearly visible. And so by taking the difference between the cross peaks, the J values can easily be calculated, compared to COSY, where many of the cross peaks are cancelling out because they have different phases. There are a lot of plus minus. So they are cancelling out. So it is much more difficult to calculate J values from COSY. Another like application of this is that suppose there is a this DNA is binding to a molecule mithramycin. So how the structure is changing after binding, that will let us know that whether binding is happening or not. So we can take the NMR of just this DNA, just the molecule, and the joint NMR of both, and that will give us an idea of how the geometry is changing with the binding. Again, here we normally use the E-COSY or sometimes CT-COSY in order to get better fine structure so that we can easily calculate the J values. Another thing is that the epsilon rotation, which basically epsilon is between this C3 prime and O3 prime. So it is along the phosphate bond connecting the phosphate. So along this bond, that is the epsilon rotation, how the energy changes. This rotation is actually very much restricted to values around 270 degrees. Away from this, the energy increases very sharply. So this is a very restricted torsion of the DNA, and it generally prefers values around 270 degrees, which have the lowest energy. Just give me 2 minutes. So far, we have calculated that we have seen that from rho z, we can get the distance between protons. So rho z is directly proportional to 1 by r to the power 6. So from the rho z intensity, we can get the interproton distances. So, and we can use these as distance restraints. So the distance between two protons, I and j, is constrained between two limits, the upper limit and the lower limit, according to the rho z cross peak intensity. Using the different restraints, we define an energy function as EF plus ENOE. EF is the intrinsic energy which arises due to the different bond distances, bond angles, torsional angles, and so on. And ENOE is the NOE potential. The ENOE can be defined as a summation of uh, a constant into, so this Rijl is the lower bound minus Rij whole square, plus again a summation of a constant into the distance in minus the upper bound, if the this u means the upper bound. So the deviation of the distance from the upper bound whole square multiplied by a constant summed up over all the protons, and the deviation of the distance from the lower bound whole square multiplied by a constant and summed up over all the protons. If all these distance restraints are satisfied, then this ENOE becomes zero. That is, the only energy is the intrinsic energy of the uh, uh, intrinsic energy due to the different bond angles and so on. So in the calculation of any structure, the goal is always to make this ENOE zero. So the structure has to be optimized again and again till all the violations to the distance restraints are removed. So all the distance criteria are satisfied, and in the optimized structure, the ENOE is zero. One procedure that is followed for that is called the torsion angle based distance geometry, or TAND algorithm. Here are 12 more DNA. So basically, to uniquely define any structure, a certain number of distances must be defined. For a triangle, 3 distances must be defined. For a quadrilateral, 4 distances must be defined, and so on. So we have certain distance constraints from the nosy intensities. Here, a 12 more DNA is shown. We start from a random geometry. We put the distance constraints, torsion constraints as obtained from NMR, and we start the optimization. During the optimizations, as more and more of the constraints are getting satisfied, the structure is going more and more towards the helical structure that we all know. And the optimization is continued till all the constraints are satisfied and the ENOE becomes zero. And that is the final energy minimized optimum structure that we have. So this is used to obtain the optimum structure from the NMR. And sometimes HS, so far we have seen nosy, cozy. Sometimes HSQC can also be used in order to get information about the base pairs. So you can see here it is the HSQC spectra. On one axis it is the hydrogen, this axis it is 13C. You can see the correlations shown by arrows here. These different kind of HSQC correlations can be observed. They can often help us determine what are the base pairs also. So now we had looked briefly into the G tetrad and how they stack in order to form the quadruplex. We will look more into the geometry of these quadruplexes now. So the G tetrad is where 4 guanines come together to form this kind of a tetrad structure. There are hydrogen bonds connecting all the G's, and each G is represented by one square here. So this entire plane formed of 4 G kinds of arrangements. The strands can all be parallel. it can be a 3+1 hybrid. So 3 strands are parallel and 1 is anti-parallel. Or it can be anti-parallel. So along the diagonal of this tetrad, along the diagonal, these 2 strands are anti-parallel, and along the other diagonal also, these 2 strands are anti-parallel. Or it can be anti-parallel. So this is anti-parallel defined as this. So why it is defined like this is because we saw along the diagonal they are anti-parallel. But if we look along this side, these two are parallel. If we look along this side, these two are parallel. So this is a kind of anti-parallel defined by 2 sets of parallel. Or it can be anti-parallel where it is up, down, again up, again down. So it is alternating anti-parallel. Another kind is that you can have a chain reversal like this. So here the strand is coming down. There is a loop here which is reversing the chain, and again the strand is coming down. So this is called a double chain reversal loop. Or it can be an edgewise loop. So the loop is along the edge of the tetrad. Or it can be along the diagonal of the tetrad, calling it a diagonal loop. The entire thing can be a monomer. So you start from here, there is a edgewise loop. Then you come here, another edgewise loop, and then you come here, there is a double chain reversal loop. So the entire association is formed from a single strand. Or it can be a dimer like this, where there are 2 edgewise loops, and so there is a dimer structure forming the entire quadruplex. Now for each G, we know there is 1 iminoproton. But if we take the spectra in D2O, the H can exchange with D and many of the peaks will not be observed. But if we take the spectra in H2O, all the peaks will be observed. So if there are 3 tetrads stacking like this, each tetrad is made of 1 G. So there are total 4 into 3, so 12 G in this whole structure. And each G has 1 iminoproton, so there should be 12 iminoprotons in this whole structure. You can see here 2 different, different associations of the tetrads are shown. So here you can have 2 parallel loops. So this is a loop which is a so if you imagine the loop as a structure, these 2 loops are parallel, whereas in this case these 2 loops are anti-parallel because one is going up and one is going down. Now if you have what is shown here is a iminoproton spectra of this sequence, where there are you can see according to the colors the peaks have been labeled. So the red G and the blue G are shown by this red and blue peaks, and the green and yellow are also shown. So since you observe peaks from both the structures are coexisting in the DNA we are observing. So for all these structures, it is the iminoproton region that we use to understand which structures are present. Here, so these quadruplexes, they can form, you can see often in these spectra, you will see potassium ions because the K plus ion will fit into the space between the quadruplex and stabilize it. Or here you can see it is Na plus, it is fitting into this space and stabilizing it. So that is the purpose of the ions. One thing is that, so you can see these are different, like, so you can see these 2 are the same sequence, AG3T, AG3T, only with different mixing time. So we might have to standardize this mix. In the lower mixing time, some peak like this G4 peak is not visible at all. Similarly, this peak is not visible at all. So a higher mixing time may be needed in the 2D spectra. 180 milliseconds seems good enough to get a good spectra. One interesting thing here is that the amino proton peaks are also observed. So why they are observed is because uh, amino protons, if we look at, so you can see the amino protons, they are locked in the tetrad, so they are not able to exchange. And so even the amino proton peaks are being observed in these tetrads. Here you can see that AG3T, these are the three peaks for the Gs in the immuno region. And with increasing temperature, this tetrad is dissociating, so the peaks are disappearing. So that is again what happens with increasing temperature. Now, G tetrad is formed with 4 Gs. You can also have an A tetrad formed with 4 adenines. Or a T tetrad formed with 4 thymines. So there can be different kinds of tetrads like this with different patterns of hydrogen bonding. There can even be heteromeric structures where you have a 8 tetrad stacked over a G tetrad and so on. So here the A tetrad is shown by the cyan color and the G tetrad is shown by the red color. So they are stabilizing each other and they can often be present in this kind of a heteromeric structure as well. These are much more complex NMRs. We don't need to go into the details of that. I'm just describing the different structures. So in this, we have covered a lot about the different DNA conformations and within the DNA itself, that how the conformations of the sugar ring can change, and with respect to the sugar ring, how the phosphodiester bond or the nitrogenous base orientation can change, and so on. Now we will cover some questions based on these concepts. The first question is asking, in a DNA double helical structure, the two strands are running in opposite directions held together by the following hydrogen bonded base pair. So here, if nothing is mentioned, we have to consider the most common type of base pairing, that is the Watson-Crick base pairing, where adenine forms 2 hydrogen bonds with thymine and guanine forms 3 hydrogen bonds with cytosine. So it's option B, A, T, and GC. So if nothing else is mentioned, we have to consider the most common Watson-Crick base pairing. The next question is asking, in the nucleic acid structure, the pseudo-rotation P angle refers to what? So here, again, if we look back, the pseudo-rotation angle refers to the geometry of the sugar ring, that is option A. The pseudo-rotation angle can vary from 0 to 360 degree, and the different conformations occur at different pseudo-rotation angles. Most common is the C2 prime endo, that is around here, somewhere between 144 to 180, and the C3 prime endo, which occurs around here, somewhere between 0 to 36 degrees. These are the two main ones that we will look at. And we know that nu is the torsion along any of the five bonds in the five-membered sugar ring. This can also be defined in terms of the pseudo-rotation angle using this formula, where tau m we know is the maximum deviation from the plane. Cos of P varies from 0 to 360 degree with different sections belonging to different orientations, plus I minus 2. I is the nu that we are looking at from 0 to 4, and delta is 144. So using the pseudo rotation angle, we can define any of the nu, that is any of the torsions in the pentose sugar ring. So P refers to the geometry of the sugar ring A. The next question is asking how RNA differs from DNA. So here the answer is A. Thymine is replaced by uracil. So we have discussed that between RNA and DNA, guanine, adenine, and cytosine are common, but there is thymine in DNA, which is uracil in RNA, and these differ that thymine has an additional CH3 group. This is very evident in NMR because CH3 group appears very much upfield shifted, less than 2 ppm. So we can understand it is thymine. There are other differences in the sugar ring. For example, at the 2' position, DNA has two hydrogens, whereas the sugar ring in the RNA, that is ribose, it has one H and one OH. So this 2' hydrogen is downfield shifted in RNA compared to DNA. But here the options are all about the nitrogenous bases. So the correct answer is option A, thymine is replaced by uracil. The next question is about bDNA. What is the handedness of the helix? What is the pitch? And what is the sugar puckering? So here, you don't have to remember for A or Z DNA, but since B DNA is the most commonly found DNA, it is best to remember the properties. So B DNA is right-handed, so automatically A and D get cancelled out. There are the pitch is 34 angstrom, so a rise of 34 angstrom per turn, and there are around 10 residues per turn. So the rise per residue is 3.4 angstrom. Pitch is about how much the rise is for an entire turn, and that is 34 angstrom, which can also be written as 3.4 nanometer. So it is option B. And you can also remember that it is C2 prime endo is the one that is um, adopted in the B DNA. The C3 prime endo is for A DNA. So for the sugar ring, it is the C2 prime endo, that is 2 prime is above the plane, 3 prime is below the plane. You can also remember the other properties for the glycosidic bond, that is this chi torsion. It is the anti, that is the 5-membered ring faces towards the oxygen, and the diameter is around 20 angstrom, and the base pair tilt with respect to the axis is only around 6 degrees, because in bDNA the base pairs are mostly not tilted, they are perpendicular to the axis. So since bDNA is the most common kind of DNA, it's best to remember these properties for the bDNA. And also there is a major groove and a minor groove. So the correct answer is option B, that bDNA is right-handed with a pitch of 3.4 nanometer and sugar puckering C2 endo. The next question is asking, nitrogen of 5-membered ring of purine is hydrogen bonded to pyrimidine in what kind of base pair? So here we look at the different kinds of base pairing. In Watson Crick, it is A with T or U and G with C. And in case of the purines, only the 6-membered ring is involved. If we have the similar base pair, that is A with T or U and G with C, but for the purines, one from the 6-membered ring, one from the 5-membered ring is involved. That is the Hoogsteen base pairing. That is what this question is asking. That nitrogen of 5-membered ring of purine, this is the 5-membered ring of purine, is hydrogen bonded to the pyrimidine. This happens only in case of Hoogsteen, because even in wobble, it is only the 6-membered ring of purine that participates. The 5-membered ring of purine participates in hydrogen bonding only in the Hoogsteen base pairing. So that is the correct answer, option C. The next question is asking that if we are getting peaks in the range 0 to 1.5 ppm, to what do they belong to? So here the question is thymidine, because such upfield shifts less than 2 ppm can only belong to the CH3 group, which is this CH3 group that is present only in thymidine. All other shifts, whether it belongs to the nitrogenous base or the sugar protons, they all occur downfield of 2 ppm. But 0 to 1.5 ppm range is very characteristic of this CH3 group in thymidine. So that is the option C. The next question is asking, for the structure calculation of nucleic acids, which of the following information is required? So here the answer is all of the above, because when we look at the formula for the structure calculation from the NMR, we have to remember that the energy function which is being minimized, it includes the intrinsic energy function, which is the energetics information C, and the ENOE, that includes the NOE information. And within the NOE itself, you will have information about the different geometry and distance constraints, as well as sugar geometry can also be part of the energetics information, that is the intrinsic energy. So the net energy function consists of two terms. The intrinsic geometry, which involves uh, the bond distance, bond angle, torsional angles, in which the sugar geometry is definitely included, and we need the energetics information of the molecule. And the NOE, which so we know NOE intensity is directly proportional to 1 by R to the power 6. So from that we get distance constraints. And based on these distance constraints, we define the ENOE, that is the NOE information. And when all the distance constraints are fulfilled or satisfied, then this ENOE becomes 0. So for structure calculation, we need all this information. So the correct answer is option D. That is all of the above. The next question is asking, how many coupling constants are present in the sugar moiety of nucleotides? So here, if we look at the sugar moiety, there are 5 coupling constants present. From the 1' hydrogen to the 2', from the 1' to the 2 double prime, from the 2' to the 3', from the 2 double prime to the 3', and from the 3' to the 4'. These are the 5 coupling constants that we observe. The other hydrogens don't really stay as hydrogens. This forms a phosphodiester bond, this forms a phosphodiester bond, this connects to the nitrogenous base. So these are the only 1, 2, 3, 4, 5 hydrogens involved, and there are these 5 coupling constants. So, and each of these coupling constants vary with change in the pseudorotation angle defining the sugar geometry. So there are 5 coupling constants present in the sugar moiety of nucleotides. So the correct answer is option A. The next question is asking which experiment is used to determine sugar geometry fine structure. Just once, yeah. So here it is cozy. So cozy is all about the fine structure. You can have you can have the so the sugar geometry can be defined by cozy. For example, if you have C2 prime endo, in this the J coupling constants, there are 5 J coupling constants and 2 of them are very small. The 3 prime 4 prime is around 0 and 3 prime 2 double prime is around 1 to 2. So these peaks are not observed. The only peaks observed are 1 prime 2 prime, that is 1 prime 2 prime and 1 prime 2 double prime. So these two, and 2 prime 3 prime. So this one. So these 3 peaks are observed in cozy, and the fine structure can be calculated. We have already seen that before, that using the active and passive couplings, the sign can be determined, and depending on that magnitude, it will depend whether there will be this kind of a crossing or there will be no crossing. So like that, the fine structure of each peak can be determined, and by multiplying them, we will get the fine structure of each of these cross peaks of the sugar ring. So to determine the sugar geometry fine structure, it is the cozy experiment that is done, which is option B. The next question is asking that cozy 45 gives a better fine structure due to what? So here the answer is intensity of the cross peaks are altered. So basically, it has nothing to do with signal to noise ratio. What happens is that in cozy, a lot of the cross peaks were getting cancelled out since they were of opposite phases. You can see there were opposite signs. So a lot of the cross peaks were getting cancelled out. And cozy 45 simplifies the fine structure. And how it does that is that the intensities are altered, hence reducing the cancellations. So it has nothing to do with signal to noise ratio. The number of cross peaks are not altered, only the intensities are altered so that they do not cancel out. So it is option B, that is the intensity of the cross peak is altered to get better fine structure in cozy 45. And finally, the question is asking about the epsilon torsional angle and what is the lowest energy for that. So if you remember this graph, you can see the epsilon around 270 degree gives the lowest energy. Epsilon is the angle between the C3 prime and the O of the phosphate. So this angle has very restricted values around 270 degrees, because that is where energy is lowest, and away from that energy rises very sharply. So that is the option D, that is 270 degrees. So that is all we'll discuss. So in this class, we have covered a lot about DNA, different conformations, different complexes, and the basics of 1D and 2D NMR of DNA. Next class, we'll discuss about proteins. So as always, this recording will be uploaded and you guys can go through it at your time.